All right. I am sorry that it took me so long to get this up here. I've been trying to find the time to video. And so I'm going to go ahead and do the maggot thing now. So what this was just us being introduced in a scientific process that happened back in the past, which was people arguing how life came about. Like, how do living things, where do they come from? And then also getting us a chance to walk through how to set up a lab report. So the maggot problem on that. So back a long, long time ago, there was a problem and people started noticing that maggots were appearing on meat. It was the meat that they were left out. Now, if I'm going to quickly, just pause the video and be able to write your stuff down and then maybe play it again. So maggots were appearing on meat and people were kind of like, where are they coming from? Because there's just some meat on the table. So where were these little living things coming from? And people had all kinds of hypotheses. So I asked the class to form their hypothesis, which will go right here. Now, not everybody, some kids know where maggots come from. Not everybody knows. I've had years where I've had no kids know where they come from. So just try your best, write down what you think, where you think they're coming from, and then I'm going to tell you what this first scientist, Reddy, what he said. So if you need to pause it, like I said, go ahead. I'm going to go ahead with what Reddy said. Reddy's hypothesis was that maggots come from flies. So this is really where maggots come from. They come from flies. But then how do you show somebody in a scientific way that that's what's happening? So I asked the class to take about 5 to 10 minutes and to either draw how you would set up an experiment or maybe write down and tell me exactly how you would do an experiment. So if you want to go ahead and pause the video right now and try to think about how would you prove to somebody that maggots come from flies? If they're like, I have these maggots appearing on my meat, but I don't know where they're coming from, and you're like, it's a fly, how would you test that? How would you show them that it's flies? So go ahead and maybe pause the video because I'm going to go on and talk about what this scientist, what Reddy said. All right. So hopefully you have your experiment done. So here's what Reddy did. I'm going to draw it out. Okay, I'm not an artist, so I am sorry for how this might look. So he had 4 containers. There's 2, and then he had 2 more. So he had 4 containers, and then here's my meat. This is my meat. Okay, it's just kind of a blob down here at the bottom. You put meat in the bottom of these containers. And then he did one specific thing, because all our tests, right? Anytime we do a scientific test, everything has to be the same. So the same type of container, same size, same type of meat, the same amount of meat. The only thing you do is you change one thing. And the one thing he changed about this experiment was he put a top. He put a cover on top of these 2. So he covered them up. He let them sit. He let them sit out. And guess where the maggots appeared? The maggots appeared right here. Okay, so maggots. And then of course these were closed up, so no maggots. So he was like, see, maggots come from flies. But there's a big problem with that. It's like, what if something other than a fly got into that container? So we had some scientists that were kind of back and forth with it, but we're going to see the whole thing play out. All right, so I did some just kind of review with the kids. I was like, all right, so we have, what we changed is the lid, and what we're measuring is maggots, right? So what's my dependent and what is my independent variable? Well, we said, do maggots depend on the lid or does the lid depend on the maggots? And the maggots depend on the lid. And then we said, okay, well, which one is our control? Well, we were testing to see if the maggots were coming from the flies, so this was my test. This is my control to show that, hey, this is what normally happens. right? So we always have these two variables, dependent, independent, and then we have our control. And like I said, we only ever want to change one thing in an experiment, and that was the lid. All right. Ready's recorded data and results. So when we're doing a lab write-up, we have our experiment, and then after the experiment, we have our data. So his data would be no lids equals maggots, lids equal no maggots. Now this is very simplistic. When you guys do your data, you may have had a table, and it may have been, oh gosh, glass number 1, glass number 2, 3, and 4, and then it may be day 1, day 2, and it may be, oh, no maggots, no maggots. Well, yes, we have maggots, and then no, 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 no. So this could also be what your data looks like. It's usually going to be in the form of a table or a graph. Okay, but I'm just writing down what he found. So once he did this, Ready's conclusion was then that he accepted his hypothesis. Maggots come from flies. All right, so once you have your conclusion, that's the end of your lab report. But as a scientist, you then publish your results. So you send it out so other scientists can look it over and be like, yeah, I think this is a great experiment. I think everything went well. Maybe that scientist wants to do the same test and see if he gets the same thing you get, or maybe a scientist looks at it and said, no, there's something wrong with this experiment. So he retests it, and then he publishes his results, and we get this back and forth with the scientific community. But that's what has to happen for us as a science community to agree on something, like a theory or something like that. All right, so what happened is we had some scientists that were like, hey, you have a flaw with your experiment. Like anything could have gotten into that. So we had this guy named Needham, and these are their pictures over here. Needham was with a group of scientists that said maggots come from like a special gravy. He said there's something called spontaneous generation. And spontaneous generation means that if you have the right conditions, then life just appears. So if I put a paper and a pencil at a desk, then poof, I turn around and poof, a kid appears, a student. Or a lot of people thought that with mice, all you need to do was put a bag of grain out and poof, mice would appear. Like they weren't born, they didn't start out as babies being born, that they actually just showed up out of thin air. So spontaneous generation, that maggots came from this special gravy. Now I don't know why people thought this, and for instance the special gravy, because obviously Reddy did not have a special gravy, but he still got maggots. But he said you had to have the right conditions, and that spontaneous generation did occur under the right conditions. So Spallanzani, another scientist, was like, okay, well, I don't believe in this, but I also think that we could do better. He was like, I'm going to prove Needham wrong. So he did use this special gravy. So I'm going to, instead of writing, I'm going to draw out what he did. So now on the bottom of this is a special gravy. The special gravy that Needham said you needed. He did the exact same experiment, right? He covered one, he left one open. Guess what? We had maggots, and then we had no maggots. So he said, see, even though there was that special gravy right there, no maggots appeared. It's not the special gravy that's doing it, it is flies. So Needham came back and said, guess what he said? He said, there's something really, really wrong with your experiment. He said, all living things need something to survive. And since maggots are living, guess what they need? When you have a lid on a jar, they have no air, they have no oxygen, and therefore the maggots could could not breathe, so he still clung on to this spontaneous generation, that maggots need this gravy, but they need it in the right conditions. So, pasture to the rescue. All right, if you've heard of pasteurization, pasteurized milk, then this is the scientist that we have to thank for that. He has done a ton for the scientific community. So what he did to put this to rest, and he was like, fine, I'm going to give you your special gravy. So what he did is he had a contraption that looked like this with this special gravy. Now he heated it, so I'm going to put like some flames up here. He heated it because he needed it to stay heated so that it wouldn't, you could get moldy and all that kind of stuff. It wouldn't go bad. So it's not like he was setting this on fire, he was just keeping it warm. So anyways, what would happen is that these little particles, like any bacteria, anything, they would come in and they would fall right here, and they could not make it up that hill to get into the gravy. But it was open to air. All right, so he let this sit like this for 1 year. For 1 year, that thing sat there and did not have any maggots. So 1 year later, he took it. So think how long it took them to convince somebody that maggots come from other living things. He cut the neck off. Here's that special gravy. So here, we never had any maggots. He cut the neck off of it, and all of a sudden, within a few days, maggots appeared. So he said, living things come from other living things. Things do not just appear out of thin air. So that kind of put that to rest. The scientific community rallied behind him, and we no longer thought, or any scientist no longer thought, that spontaneous generation was a thing. So this was just a quick little review of like kind of, this is biology, this is the study of life, so just going through something that's living, how living things come about, but also walking you through a good lab write-up and how it's set up. All right, the last thing I did with them that day is actually go through what makes a good lab. Let me find it here. Did I not post it? I don't think I posted it, so I'm sorry. Yeah, I should have posted it here. So let me go and look right here. I'll clear the ink. Let's go back down to normal size. That. And I'm going to look for the lab. Lab report right there. All right. So this is a sample lab report. I've got them in school. I'll post it on Google Classroom. We will use this soon, next week. So what we have here is we have a purpose. You're going to tell me why you're doing the lab. So I give you an example, like, okay, if I were going to test how water affects plant growth, I give you examples how I would write my lab as we go through. You have your hypothesis. Once again, you're getting 5 points for a title and a name, but your hypothesis. I will, in class, I've really been going over if-then, how that looks. So if sunlight causes plants to grow, then the more sunlight the plant, the taller it will be. My materials, I talked to them about being descriptive and specific. Don't just put water down. How much water did you use? Don't just put down that you need cups. How many cups did you need? How many seeds did I use? The procedure, be descriptive. So instead of just saying place 25 or place potting soil in all 3 cups, I'm telling you exactly how much. Place 25 grams because we need everything to be the exact same. Don't use the word I or we. Don't say I placed 25 grams, just place 25 grams of potting soil in all 3 cups. You can see where I said place 5 seeds. And I don't say just into the potting soil because I could go all the way to the bottom. I could sit it on top of the potting soil. I told you exactly where I put it, 2 centimeters in. All right, so just being very specific. How much water did I get? 3 milliliters. All right, my data. This might be a sample of my data, and that's all I put. I'm just putting this chart. You might have a graph or a chart and a graph, but that's it. I'm not going to tell you anything. I'm not reporting on my findings in the procedure. I'm not reporting them here in the data. I do that in the results.
I'm going to explain what this is telling me. What this is telling me is plant 1 had some water and had growth of 0.7 centimeters. Plant 2 had the most water and had a growth of up to 2 centimeters. Plant 3 had no water and had no growth. The graph shows how fast the plants grow when they have more water. All right, then my conclusion. This is where people tend to lose the most points, and that's just because there is a lot going on. You've got to summarize your experiment. Plant growth was tested by giving seeds different amounts of water. Did I accept my hypothesis? My hypothesis was correct. The plant with the most water did much better than the other 2 plants. Explain why my hypothesis was correct. Identify any strength or weaknesses. A weakness in the lab is human error. It was hard to read the ruler when the plants had not grown much. Were you limited? The lab was limited in that there were only 15 seeds. Maybe I should have done more. I would have had more seeds. I could have done more trials. Do you have any recommendations? Let's hear. Unlimited could be carried out. In future tests, I would see if too much water affects plant growth also. And then were there any sources of error? There were no errors and everything went according to plan. If something gets messed up during the lab, this is where you would tell me, and that's okay. You're not going to lose points. Don't worry about that. But I want to know if something happened, don't lie about it. Just report it and you still get the points. All right, that is it. If you need anything at all, let me know. Have a good weekend, and I hope to see you soon.